Welcome all. Thank you very much for coming today and this evening for this really cool, and I must say, really important event, as will become clear to those of us who are not working on ancient diseases. Megan Michael's paper is really quite important. It marks a watershed in our ability to study one of the most devastating diseases in human history in all likelihood, judging by what we can see of it today and what we can see in the genetic inheritance that so many of us carry in our bodies of debilitating diseases which evolved positively under the positive effect of doing very bad things to us, but being able to procure a limited defense against the disease of malaria. The Science of the Human Past, as many of you know, is dedicated to bringing together the humanities and the sciences and to discover completely new data about the human past, and to do so working in groups, in teams that bring together students, graduate students, postdocs, from the freshmen in our classes in the Science of the Human Past courses that are spread throughout the departments and programs of the university, all the way up to the fantastic postdocs and that we have the pleasure and privilege of working with. This is our steering committee, where the Science of the Human Past is really just a network. We have no bricks, we have no building, we barely have an office, but we try to do a lot with a little. And it's a network of research groups, of seminars, of laboratories, and of individuals from across the university, as you can see from the steering committee. And some of the most important parts of our steering committee meetings is simply talking about what we're working on to each other, because we don't get a chance to see each other. The Science of the Human Past is the platform within Harvard that, thanks to the support of President Drew Faust, we were able to do something which was unthinkable at the time, and that was to enter into a collaboration with another institution. And in this case, with the Max Planck Society of Germany. The Science of the Human Past is the Harvard partner of the Max Planck Society in a virtual research center that we had the privilege of creating with Johannes Krause in 2016. It is, again, completely virtual on our end. They have lots of bricks over there in Leipzig, but we have us, which is not bad. And this virtual bridge across the Atlantic brings together the complementary powers and resources of Harvard University with our tremendous programs in the humanities and the social sciences and the sciences, and the amazing technology and focus and resources of the Max Planck Society, and particularly the Max Planck Institute for Evolutionary Anthropology, of whose Department of Genetics Johannes Krause is the director. Johannes would be here with me, but he's traveling and can't get access to the internet, so he can't join us even virtually, but his thoughts are with us. This is the College of Directors of the Max Planck Harvard Research Center for the Archaeoscience of the Ancient Mediterranean. Quite a mouthful, but it was better than some of the things that had been proposed at the time, I can promise you. Many people pronounce it MOM. As some of you know, I tend to pronounce it mayhem, which is not entirely inaccurate. We have 2 graduate students. Now we actually have only 1 graduate student, because 1 graduate student has done what all graduate students are supposed to do, is the main purpose for going to graduate school, which is graduate, get out of graduate school. And Megan Michael has done that gloriously with her dissertation, results of which we're going to be here about. Reed Morgan is with us and is deep into his work in the archaeogenetics of the fall of the Roman Empire in the Mediterranean basin. So you see the cast of characters. Professor Warner is going to be speaking with. Professor Reich has a family event that is keeping him from us. We have 3 magnificent postdocs here at Harvard who are working on exploring the archaeogenetics of the human past. We have a geneticist in Alison Barton who is teaching archaeology to, he's teaching archaeogenetics to Dr. Vicky Moses, who's a wonderful Roman archaeologist, and inversely teaching archaeology to Alison Barton, and of course Solène Troadec, who's leading our investigation of early medieval France. When we set out with the beginning of the Max Planck Harvard Research Center, we were focused on 2 projects: migration, the peopling of the Mediterranean basin, not from modern DNA, but going back to ancient DNA, to the skeletons of the people who actually moved into the Mediterranean. Why? Because we surmised, and it was only surmised at the time, that there'd been a lot of migration since the times that we were interested in, that the modern genomes do not tell us who was in a place 2, 3,000, 4,000, 5,000 years ago. So migration was one aspect of our focus on ancient DNA, and the other was the diseases that these people suffered, reconstructed not from the very scattered and sporadic and often ambiguous written evidence, which only begins in any case rather late in human history, but from the DNA and other molecules of the pathogens themselves. We've had some really cool successes in recent years. Most recently, some of you will have attended in the spring the presentation of our results on migration in the Roman Empire in the provincial capital of Imanacium on the Danube in April. That's how far we've gotten. We started out with the Epipaleolithic, and we have now reached the fall of the Roman Empire, which is pretty cool in 8 years, I think. Plague was a big item, controverted when we were starting this, what the pathogen even was. And now we have 10 genomes from the Justinianic pandemic that are published. More to come, I promise. And just in 2022, Maria Spirou led the discovery and identification of the immediate source of the Black Death in Europe in a Syriac Christian trading colony diaspora on the Silk Road in Central Asia. So great progress with one disease, but there's another disease that has played a tremendous role in human history, which has been recalcitrant to the efforts of archaeoscientists, archaeologists, and historians to discover it, and that is bad air disease, malaria, as the ancients called it, because they saw that it came to be in places where there was stagnant water and ponds, and so it must have been bad air. It's been exceedingly difficult to study in the present, to look in the present. You can see what were reported to me as recent results in terms of vaccines today. It is a terrible disease, as many of us may know from human experience all too directly. But you say, so what? We're in America. No, malaria is over for us, so it's other people's problems. Well, that's true, and it isn't true. Malaria was eliminated in the United States only between 1946 and 1965 in the continental United States. I myself, as a child growing up, had to live with a father who had periodic terrible outbreaks of fever and retreated to his bed and stayed there for 2 or 3 days at a time. He had acquired that in the South Pacific between 1944 and 1945. And then, beyond the present day infections that are terrible around the globe, is the fact that so many of us carry within us the genetic burden of millennia of efforts of our bodies to fight off this terrible disease in the form of debilitating diseases such as sickle cell anemia in Africa and beta thalassemia around the entire Mediterranean basin. These are diseases that are genetically transmitted. If you get one copy of them, you're basically okay. You get two, you've got a terrible and short life ahead of you. And they're so terrible, and yet evolution preserved them because they gave us a shot against malaria, of resisting malaria in better fashion, to put it in the simplistic terms that a historian understands. So this is a big deal, and it continues to be a big deal in the world. The ancients observed it. They observed the periodic fevers that came with it, which are now understood in terms of the life of the microbes that cause it. But the records of the ancient physicians are very, very rare, and they're very sporadic. So we can see it, once every 200 years in one place in the entire Mediterranean basin. That's not something that allows us to construct an epidemiology and to weigh the impact of this disease on human beings. So what difference will it make? Now that for the first time, thanks to Megan's work, we're going to be able to reconstruct systematically the presence, the degree, the type of malaria and its impact in particular areas over time and space. We're going to understand why different regions rose and fell at different times if malaria was at play. We're going to be able to see for the first time the demographic impact of this terrible disease. We're going to be able to see the economic consequences of populations that labor against this terrible demographic regime of malaria that's been postulated but impossible to demonstrate. We'll be able to detect economic impact, and even in the cases of the migration of different types of malaria into new environments, climate change. proxy for ancient, medieval, and early modern climate change. It has been argued long ago, over 100 years ago, that malaria was a disease that unmade Greek civilization at its apex. The question was raised whether the insect that conveyed it defeated the conquering legions of Rome. And in 1907, a distant forerunner of the science of the human past, W.H.S. Jones, got together with the man who won the Nobel Prize, Ronald Ross, for discovering that malaria is transmitted not by air but by insects, to write this book on malaria in ancient Greek and Rome. Very careful, very impressive work, and should have been the beginning of the science of the human past. The work was trashed by his classical colleagues. Historians have no business reading biology, trying to include biology in history. That has nothing to do with history. We should read Greek text and reconstruct the past from that. I love Greek text, but they don't tell us everything. And poor Jones went on to have a not distinguished career, an okay career, but not a distinguished career, and no one would touch the subject among historians and classicists with any conviction for almost a century. Ross and the biologists in general welcomed the results. Said, this makes sense to us. Very good hypothesis. Let's continue. Lately, there's been a resurgence of interest and work on it. Two particular books by one of our commentators have given a lot of new light to the impact of malaria on the Roman Empire, in Fate of Rome, and then globally thereafter. Valiant colleagues have continued the laboratory work to try and get this recalcitrant organism to yield its genetic secrets with very mixed results, as they are the first to admit, and with in very cautious publications, starting with Robert Solaris in 2001 and then 2007, the work of Stephanie Marciniak in the laboratories of Henrik Poinar and Tracy Prause, and most recently, Alvi Lufuma-Mbuaka, who has done a comparative study of the impact, the use of different types of evidence of malaria in a Roman-Egyptian population. But until the paper that Megan Michael led, there was no robust way forward. There is now. She has delivered the first elements, and they are just the first steps towards what is going to come, and which is going to be very, very cool. We're going to start off with a presentation by Dr. Michael about her paper, her work. We're going to follow up with Dr. Michael, by the way, just defended her dissertation, as I said, just a couple months ago. She got her BA at Oberlin. She exemplifies how the complementary resources of Harvard and of the Max Planck Institute can come together to create the best conditions for really pioneering research. She's now a postdoctoral fellow in Professor David Reich's lab. We're going to have a response from Diane Worth, the Richard Stong Professor of Infectious Diseases at the Harvard Chan School of Public Health. Dr. Worth got her BA from the University of Wisconsin at Madison, her PhD at MIT. She is a leading expert on malaria today. Her lab studies the elusive molecular biology of protozoan parasites to discover parasite-specific interventions against them, focusing on functional analysis of genes important for parasitic virulence, emphasizing the problem of drug resistance. We'll have three very distinguished commentators, starting with Professor Kyle Harper, who took his BA at the University of Oklahoma, his PhD here in 2007. He holds the Blankenship Chair of Liberty, in History of Liberty. He's a professor of classics and letters in his alma mater, Oklahoma. Incredibly prolific historian. Graduate students, listen to this carefully. Since he got his PhD in 2007, 4 big books, prize-winning books on slavery in the later Roman Empire, later Roman world, redefined the subject, Christian transformation of sexual morality, the fate of Rome, and plagues upon the earth that you've seen, and a 5th book in progress, The Last Animals, A History of Humans and Other Animals. We will be followed by Professor Edward T. Ryan, MD, who is the Director of Global Infectious Diseases at Mass General, a professor in the Harvard Medical School and the Harvard Chan of Public Health. Professor Ryan got his BA from Princeton. He did his MD at the Harvard Medical School and his residency at Mass General. He also studied at the London School of Hygiene and Tropical Medicine. He served as an expert in multiple capacities at the Institute of Medicine of the National Academy of Sciences, the CDC, the NIH, the American Society of Tropical Medicine and Hygiene, has written more than 350 articles and book chapters on global infectious diseases. And last, but by no means least, we'll conclude with our own Professor Christina Warner in the archaeology wing of the Department of Anthropology. She had her BA from Kansas, came here and did her PhD in anthropology, completed in 2010. After stints at the University of Oklahoma and the Max Planck Institute, she's returned to Harvard, where she became a full professor earlier this year. She remains a group leader at the Max Planck Institute in Leipzig and is one of the directors of MOM. Her publications and prizes are amazingly diverse. I couldn't even begin to enumerate them, but they're strewn across nature, cell, PNAS, science, Latin American antiquity, etc. She's a trailblazer in new methods in ancient microbes, ranging from dairying its origins to the outbreak of an epidemic in 16th century Mexico, to the Himalayas population history, and my favorite, how a piece of Afghanistan got lodged in the teeth of a medieval German nun. Her big multidisciplinary project on Mongolia combines archaeology, ethnography, and microbiomes to understand the origins of dairying and human adaptations to new foods. And a tip for all of you, Professor Warner and the Science of the Human Past are going to do an event in March at the Peabody Museum with an exhibition about this big project. So more fun ahead. So our plan is to start with Professor Michael. It's high time to hear from her and stop hearing from me. Megan, take the floor. It's all yours. Thank you, friend.
Okay, can you all hear me? Okay, great. First of all, I'd like to thank Professor McCormick for that fantastic introduction, and to everyone here for coming to this event, and especially to the correspondents who are going to talk after me. And so today I'd like to present some data that we published earlier this year in a paper trying to explore the history of human malaria using ancient DNA. As we already heard, malaria is a disease caused by a eukaryotic parasite of the Plasmodium genus. And today I'm going to focus on two species in particular, Plasmodium falciparum and vivax, which cause the vast majority of morbidity and mortality in human populations today, and probably also in the past. Malaria has a really complex life cycle, the details of which aren't super important for us today. Except for the fact that we know that all of these species are transmitted by female mosquitoes of the Anopheles genus. Malaria is also a major public health issue today, as we've heard, with over 600,000 deaths annually due to malaria. And we know that even though this burden is huge today, it was probably even worse in the past. So here in this map in purple, I'm showing the distribution of malaria parasites in the year 2002. And if we take a look back over the last 100 years in this animation, you can see that a century's worth of public health and eradication campaigns have drastically reduced the regions in which malaria is endemic today. And in the year 1900, malaria was actually endemic across a full 50% of the world's land surface, reaching up into these more northern latitudes, as Professor McCormick also mentioned. So for this project, we were interested in taking a glimpse into the history of these past parasite populations to try to understand how malaria attained this global distribution, what impact these parasites had on human populations in the past, and ultimately to try to understand something about the evolutionary processes via which these ancient parasites evolved to be the pathogens that cause disease in humans today. But first, just a quick look at the sources of information that we can use to try to understand disease in the past. And a major source of information about malaria in history is textual sources. And we've heard a bit about textual references to the cyclical fevers that cause malaria in Greek and Roman medical literature. However, we face a major limitation when we're dealing with texts, in that many regions and time periods in the past lack writing, and those are completely invisible to us if we want to study malaria in those time periods. We can also turn to the archaeological record and to paleopathology to try to understand the distribution of different pathogens in human populations. And we know that certain pathologies, such as cribrorbitalia and parodic hyperostosis, are often associated with anemia in past human populations. But anemia is a condition with many potential underlying causes, so this isn't a very specific marker of malaria in the archaeological record. Key methodological advancements in the field of ancient DNA over the last decade or so have begun to transform the types of questions that we can ask about infectious disease in the human past. And now we can not only obtain ancient DNA from human bones and teeth, we can also even obtain DNA from sediments and from cultural objects. But most importantly for this talk, studies over the last 10 years or so have shown that teeth from humans from archaeological sites can often preserve traces of bacterial and viral and even eukaryotic parasite DNA that were present in the bloodstream of people at the time of death. And this provides a novel source of information about these infectious diseases in the past. But studying ancient pathogen DNA is a really challenging endeavor. Not only do we have to deal with the fact that this DNA is damaged and highly fragmented, we also have to contend with the fact that ancient microbial DNA is typically present in extremely small quantities. So in this little cartoon, the vast majority of DNA that we extract from this sample is from environmental contaminants, things like microbes in the soil, whereas a smaller fraction comes from the humans whose skeletal remains we're actually studying. And the endogenous malaria DNA typically constitutes tiny fractions of a percent of the DNA that we retrieve from a sample. So this makes identifying samples that harbor this precious ancient malaria DNA sort of like trying to find a needle in a haystack. And partially because of these limitations, work so far on ancient malaria DNA has been very challenging. We already heard about the landmark study by Marciniak and colleagues that succeeded in retrieving some fragments of mitochondrial DNA from 2 individuals from the 1st to the 2nd CE, southern Italy. But this study also faced some limitations. So this DNA was only from uniparentally inherited organelles, and it was also too low coverage for analyses beyond species identification in those individuals. Aside from those samples, the only other ancient data that we have is actually more historical than ancient. It's only about 70 years old, and it comes from blood slides like the one you see here that was produced in 1944 in the Ebro Delta region of Spain. And these blood slides are incredibly valuable sources of information because we know that malaria in Europe today is now eradicated. So they provided a first glimpse into what these historical malaria parasite populations look like in terms of their population genetics. However, 70 years is not that long, and we really wanted to take a glimpse further back into the past to look at population affinities over time. So one of the most important methodological tools that we use to overcome these limitations of working with ancient pathogen DNA is hybridization capture. And hybridization capture is a technique that uses synthetic DNA baits, which are complementary to the genome of the organism that we want to study. And these can actually bind to and physically fish out DNA from our species of interest, making it economically feasible to sequence whole genomes from bacteria, viruses, and in this case, eukaryotes present in archaeological individuals. So for this study, my colleagues and I designed 2 separate hybridization capture arrays targeting the mitochondrial genome of human infecting Plasmodium species, as well as a separate array targeting the nuclear genomes of Plasmodium falciparum and vivax. And aside from using a hybridization capture approach, a second development in the field that really paved the way for this and other studies of ancient pathogens is just the sheer amount of data that's been produced from ancient individuals, primarily for studies of the human ancient DNA content. And so for my PhD work in collaboration with colleagues at the Max Planck Institute for Evolutionary Anthropology, I was able to take advantage of a large number of ancient individuals from over 3,000 archaeological sites around the world who were collected and sequenced primarily for analysis of their human DNA content. And there are certain biases in this data set, of course. We see that Europe is pretty well represented, and a lot of these samples are from relatively recent time periods, but we're starting to fill in the gaps in this map. And importantly for me, these data sets represent a really large and largely untapped resource for studying ancient pathogens in the past. So we were able to go back to these data sets and computationally sift through them to try to identify individuals harboring fragments of malaria DNA to which we could apply our hybridization capture. And using this approach, we were able to obtain genome-wide mitochondrial and nuclear data from 36 malaria-infected individuals from around the globe. And these include cases of vivax, falciparum, and also Plasmodium malariae, as well as several individuals who were infected with more than one Plasmodium species. Most of these samples came from teeth. As I mentioned, teeth are a really good source of ancient pathogen DNA. But we also identified traces of malaria DNA in several bones from the skull and a couple of calcified nodules. And we were really excited that the temporal distribution of these samples is pretty large. So we were able, in particular, to identify 3 cases of Plasmodium vivax going back to the 4th millennium BCE from across Europe, which gives us some insight into the time depth of this parasite in that region, far prior to when we have written records. So there are a lot of stories that we can begin to tell with this data set, but I'd like to focus on 3 case studies that we discussed in our paper, and which I think really nicely highlight the link between the spread of malaria parasites and human mobility at global and regional, but also at more local scales. So to start from a global scale, one of the questions we were really interested in trying to address is the transmission of malaria into the Americas. And this has been a topic of some debate in the historical literature, with people hypothesizing that either falciparum, vivax, or both might have been transmitted to the Americas during the colonial period, either from European colonizers or as a result of the transatlantic slave trade. Other people have suggested that vivax, which is more cold adapted, could possibly have arrived with the first peoples of the Americas, whereas other people suggest that malaria might have been transmitted via contacts with Oceanian seafarers, potentially even prior to European contact. So we were really thrilled to be able to collect some data sets and generate some data to address these questions. And in particular, we retrieved relatively well-preserved Plasmodium vivax DNA from an individual from the site of Laguna de los Condores, which you can see over here in the territory of Peru today. And this site sits on the Amazonian side of the Peruvian Andes at relatively high altitude, and it had really impressive preservation of organics, which may have played a role also in our recovery of significant amounts of vivax DNA at that site. The individuals associated with the Chachapoyas culture, and when we radiocarbon dated them, they fall to right around the period of European contact. So we use this data set in combination with really rich modern Plasmodium vivax population genetic data that's been published by projects such as the Plasmodium vivax Genome Variation Project, to actually try to understand the genetic affinities of this peri-contact vivax strain to modern populations. So here I'm showing a principal components analysis, just including the modern data, and you can think of a PCA as sort of a genetic map where modern strains that are more closely related to each other plot together in PCA space. And here I've colored these genomes by their geographic region of origin. So we can see that principal components 1 separates out modern Plasmodium vivax populations from the Americas, Western Asia, and Africa from Southeast Asian and Oceanian strains, whereas PC2 separates out these populations from Oceania from Southeast Asia. And what we did was project our ancient data sets onto these axes of variation. And I'd like to draw your attention to a few observations. So the first thing we noted was that when we project our Latin American strain onto this modern diversity, we see that our peri-contact Peruvian sample falls directly on top of the diversity of Latin American vivax today, suggesting a really close relationship between these strains 500 years ago to strains still circulating in the Americas today. We also wanted to dig into this a bit further, so we used statistical methods to compare our peri-contact Peruvian genome to subpopulations from all of the countries shaded here in green, And we found that this ancient sample is most closely related to vivax that still circulates in Peru. For us, this was really exciting because it suggests at least some continuity of genetic ancestry across 500 years of human history, and could imply that there was an establishment of an endemic malaria focus in that region soon after the period of European contact. So to go back to our original PCA, I want to point out a second observation. When we projected samples from Europe, from Belgium, Austria, as well as the Ebro blood slide that I showed you before, we found that these are really closely related, or that they fall very close to our modern and ancient Latin American lineages. And this observation was borne out when we constructed a phylogeny using these strains. We found that a clade formed by all of our European lineages falls basal to the modern and ancient Latin American vivax. So to us, this strongly suggests that Plasmodium vivax in the Americas was probably transmitted from European colonizers during the contact period. While I won't get into the details of our Plasmodium falciparum population genetic analysis, we found a rather different story for this species. And we know from analyses of modern strains that falciparum in South America is really closely related to strains circulating in Africa. What we can say from the ancient data is that the European strains look quite different. So we think that while vivax was probably transmitted from European colonizers, it's more likely that falciparum spread to the Americas as a result of the transatlantic slave trade, suggesting rather unique histories for these two species in the Americas. So to zoom into a regional scale, we were really lucky to collaborate with a large number of colleagues on this site of St. Rombos Cathedral in Mechelen, Belgium. This is a cathedral cemetery with over 3,000 graves that have been excavated from 3 distinct archaeological phases spanning the 12th to the 18th century AD. And what was really interesting for us is that the use of this cathedral cemetery spans the period of use of a Spanish military hospital, actually the first permanent military hospital in early modern Europe, which was constructed directly adjacent to this cathedral cemetery. So one question my colleagues were interested in was where people who were treated at the military hospital and died may have been buried, and we thought they might have been buried in that cemetery. So we were able to combine analyses of human and pathogen population genetics to shed some light on the people buried in this cemetery and the diseases that they might have suffered. And in collaboration with one of my colleagues, we identified what we think are 2 distinct subpopulations present in this cemetery. So without going too much into detail, we found that the earliest transect, the one predating the use of the military hospital, had about half men and about half male and half female individuals. And when we look at them in a principal components analysis of modern West Eurasian human genetic diversity, they form a tight cluster overlapping where we expect people from Belgium to fall in PCA space. On the in contrast, the mid and late transects are almost exclusively male. And when we plotted them in PCA, we found that they form a much more diffuse cluster overlapping populations from northern Italy and Spain and Mediterranean regions, which we know the Habsburgs were recruiting from northern Italy and Spain during this period to fight in the Netherlands. So we think that this is probably consistent with these individuals being soldiers in the military at that time. What was really cool for us was that we found a difference in the malaria infections in these individuals. So in the early transect, which we think were probably local people, we found 2 cases of Plasmodium vivax. However, in the mid and late transect, we found many more cases of malaria and also different species of malaria. Most interesting for us, we found several individuals who were co-infected with Plasmodium falciparum, a species which was not thought to be endemic north of the Alps during that time and requires higher temperatures to sustain ongoing transmission. So altogether, we think that these individuals might have been soldiers and that movement of troops in the context of an ongoing military conflict might have played a role in the introduction of falciparum into this region. The last case study I'd like to very briefly mention is a case study of individual mobility from the site of Chakopani in the territory of modern-day Nepal. And this site dates to around 3,000 years ago. and is situated at high altitude at about 2,800 meters above sea level. We were really astonished to find a case of malaria at this site because this region is far too cold and dry for either the mosquito vector or the parasite to thrive there today. When we talked further with the archaeologists and Tina, who was involved in this project, we learned that there's actually artifactual evidence from the site for trade contacts between these high-altitude populations and lower-altitude populations, probably from the territory of modern-day India. So we think that this may be a case in which we can infer individual mobility from infectious disease status, telling us something about the life of this individual who lived 3,000 years ago. So I think I'm over time, so I'd like to close just with the sort of conclusion that I think that ancient malaria genomes provide a unique look into interactions between human populations, pathogens, and the environment through time, and that I hope that this study is only the beginning and that we can learn more about the evolution of malaria through the use of ancient DNA. And I would, before I end, like to thank the very important people who, without whom this work wouldn't have been possible. So Johannes and Alexander were the primary advisors of this work, and my colleague Irini, who helped me a lot with the human population genetic analysis, and all of the many colleagues from archaeology, history, evolutionary biology, and all aspects of the science of the human past who made this study possible. Thank you.
Thank you, Megan. We'll reserve the questions for the end. We'll have a general discussion in which hopefully everyone will be involved, and we'll have a lot of fun. Professor Wirth, if we can invite you to come to the podium.
Well, Megan, congratulations on that really interesting piece of work. I think it really is groundbreaking. What I'm going to do is talk to you a bit about the situation with malaria today. I have a few slides, but I also will perhaps go through them quickly. Okay, so as we speak today, there is an effort by the global health community to eradicate malaria, and many countries, in fact, have eliminated malaria. So, you know, this is something in modern times that was quite recently called for or highlighted by the Bill and Melinda Gates Foundation, and they led what I would say is the renewed effort. There was a previous effort to eradicate malaria in the middle of the 20th century, but that failed, and we'll see kind of results of that in a second. So the plan really is to eliminate this completely preventable and treatable disease, yet it has been with us since ancient times. So why is that? Well, let's look at the situation. This is kind of the cases on, I think, the left and the deaths on the right of the current situation with malaria. So there are, 250 million cases of malaria and over 600,000 deaths. In fact, since COVID, these numbers have been going up a bit, and in fact, progress has plateaued. We make quite good progress in the early part of the 21st century, but progress has really stalled. In fact, as we speak today, this is animated, let's keep going. Over 70% of the malaria is in 4 countries of the world. Nigeria, 25%, DRC, Tanzania, and Mozambique. So it's a very concentrated place where this disease or where these parasites exist. But of course, we've been making progress. As I said, many countries have eliminated malaria. And if we look over the past 20 years, these are in different colors. I think the fewer than 10 cases is in blue, and fewer than 10,000 cases is in yellow. And you can see the numbers of countries that are approaching elimination has been increasing, not exponentially, but kind of steadily. And so there's hope for this, but of course there are challenges. Just to In fact, I should have changed this slide. Capo Verde, when I made this deck, was the last country certified. But since then, Egypt was certified about a month ago. So we are making progress. But of course, there remain challenges. I mentioned COVID. So this is comparing 2019 to 2022, the last World Malaria Report that we have. There'll be another one coming out in a couple of weeks. And you can see the number of cases has plateaued or gone up slightly, but within the confidence intervals. So why is this, and why has the parasite been around for so long? Well, I would say the answer is resilience. Okay? Both the mosquito and the parasite are resilient. And what you see on the right-hand side of the screen is, sort of these efforts to get rid of the disease in many different places. I think this is in the African subcontinent, but this is true around the world. Disease goes down to nearly zero, and then the country says, good, job done, and you can see rebound in almost every situation. And that's because it's very hard to interrupt transmission, and the basic R0 of this disease, particularly of Plasmodium falciparum, the major parasite in Africa, is greater than 100. So for those of you who aren't infectious disease specialists, I'll ask Ed, what's the R0 of measles? Yeah, right. COVID was 2 to 3. So this explains in part why it's very hard. You have to get rid of all malaria, or you will face this rebound. And I think, speaking to, sort of the more recent history, evolution of this parasite is ongoing. Response to, and the mosquito, response to the measures that we have are essentially failing because the parasite or the mosquito can actually develop a mechanism to escape. So it's really evolution in real time, including behavioral evolution, mosquitoes changing their biting habits to avoid indoor biting because that's where insecticide-treated nets are. So it's an ever-changing target, and I think that represents the problem. I'm just going to cite some work from a colleague of mine, Dan Neefsey, who's in my department, speaking directly to Megan's observation about Plasmodium vivax in the Americas coming from multiple sites. Dan has done a lot of analysis in that area, and indeed the population structure in current Plasmodium vivax reflects perhaps the now ancient and no longer existent European parasite population. So I think this work has not only great historical value, telling us the history, but also can inform us about what's going on in present day. So we're back to the challenges. So here's the situation with insecticide resistance. This is resistance to pyrethroids. It is everywhere that malaria is transmitted. You can find Anopheles species. All species, in fact, that transmit the disease show some level of resistance. The parasite has been able to delete the genes that encode the proteins that are used for the most commonly used rapid diagnostic test. It's amazing. And then finally, drug resistance, also invasion of new species into Africa. So Anopheles stevensii is an Asian and Middle Eastern mosquito, but it is now present in the Horn of Africa, and of course, we anticipate spread throughout Africa, particularly in urban areas where Anopheles gambia, the more common Gambia complex mosquito, doesn't thrive very well. It's not adapted to urban environments, but Anopheles stevensii is highly adapted. And finally, drug resistance. This is just the situation with artemisinin resistance. artemisinin combination therapy was introduced in widespread use, first in Southeast Asia, where resistance appeared, and now independently appearing in Africa, particularly in Eastern and the Horn of Africa, where we now have situations where, and this is just data from Uganda, just to show you, this is a publication from Phil Rosenthal's group, showing the presence of mutations in the gene that mediates the artemisinin resistance being at less than 1% in 2016, rising to as much as, in 2022, as much as 20% of the population. And we're just back from the American Society of Tropical and Hygiene meeting last week. There are regions of, this is Uganda, where you see 40 or 50% of the parasites in particular populations carrying this marker for resistance. Fortunately, because it's combination therapy, resistance, the appearance of the resistant marker doesn't lead to failure, but we, of course, can't sit back. The WHO, I chair the Malaria Policy Advisory Committee for WHO, and we have recognized this problem first in Southeast Asia, but now in Africa, and there is a plan for countries to introduce multiple first-line therapies to begin targeting vector control, but I think this is going to be quite the challenge. There is some, I think this was mentioned earlier, there's hope on the horizon. In 2021, the WHO recommended first the RTSS, and then in 2023, the R21 vaccine. These vaccines are very similar, targeting the same protein in the parasite, and they provide partial protection. They are targeted at infants, and the outcome is reduction in overall infant mortality. And you may not think that 13% is a large number, but this is the reduction of infant mortality or childhood mortality that occurred with oral rehydration therapy. So this is, from a public health standpoint, a very big advance, but it's clearly not the magic bullet. These children still get malaria, and this doesn't inhibit transmission. There's a very active drug development pipeline, so that lots of things coming. We just heard of the completed phase 3 trial of the next drug on the market, a combination of a new chemical entity that targets a new pathway in the parasite with an existing drug in the current combination, lumefantrine. We're clearly worried about potential resistance carryover, but that is the next product, and it will be out, we hope, in 2026. Also, I think it's very important to recognize that climate change is going to change the picture for malaria. It's not clear how, but it will affect mosquito breeding habitat. It will affect population migration. My colleague Marcia Castro is here, really interested in these issues and how they affect the overall evolution of the parasite and its impact on global health. I just want to make one comment about the Yaoundé Declaration. In March of last year, the ministers of health of the 10 high burden, high impact countries, these countries that have the high burden, high impact countries in Africa, there are 10 of them, have 95% of the malaria of the world. And these ministers came together to declare that they would make malaria a focus. I'm going in a few days to Nigeria for the first meeting of the, accelerated malaria elimination program for Nigeria, the country with 27% of the global. So Minister Pate, who used to be a member of Marcia's department, has taken this on and really is focusing his efforts on reduction of burden and with a goal of elimination. So I'll end. with what we're all here to talk about, I think. It's not the strongest of the species that survive, nor the most intelligent, well, if microorganisms can be intelligent, but the one most responsive to change. With that, I'll end. Thank you.
Thank you, Professor Wirth. Again, we'll have questions and discussion as soon as we conclude the comments of our panel of experts. And now we will hear from Professor Kyle Harper, a historian and archaeologist, who will take us to a more remote past perspective on this extraordinary phenomena in human history.
Thank you, Mike, for inviting me. Thank you, Megan, for this fantastic paper. I'm going to talk a little bit as a historian about why I think the results of this paper are so interesting and so important, and the contribution that archaeogenetics of this important pathogen is making to our understanding of the human past. In the last decade or so of archaeogenetics, there's been really revolutionary new insights into the human past, but if we're being honest about pathogens, one pathogen tends to hog all the attention. Yersinia pestis, plague, it's the cool kid on the block. Plague is pretty fascinating. It's sexy. I get obsessed with plague more than most people. But if you really twisted my arm as a historian of infectious disease and said, letting caution go to the wind, what infectious disease has probably killed more human beings than any other? You would want tuberculosis on the short list, but the malaria diseases would be, to my mind, right at the top of the list as the most destructive diseases shaping human history. And they are still devastating tropical diseases. In the past, they were not limited. Certainly, the vivax parasite was not limited to the tropical region. So just the sheer significance of the contribution that ancient DNA can make to understanding what is probably the single most devastating infectious disease in terms of its impact on the suffering and early death caused to our species. Malarial diseases were far more widespread in the even relatively recent past than we are apt to remember. I just love this image of Charles Dickens traveling from Cincinnati to St. Louis, and he was being a little bit snobbish in 1840s, and remarking on the undrained interior of America that it was a breeding place of fever, agony, a dismal swamp on which half-built houses rot away, a hotbed of disease, an ugly sepulchre, a grave uncheered by any gleam of promise, a place without one single quality in earth or air or water to commend it. His Airbnb must have not been very nice the night before. But the ague is a term for intermittent fevers, and certainly he was struck by the prevalence of this disease even in the interior of America. But it's not just suffering and death, it's the social consequences of this disease that are really unlike any other. Here's some data that I put together that reflects the differences in literacy at the beginning of the industrial period in Italy and its association with the malaria burden. Greater malaria burden is really strongly associated with important outcomes of human well-being like the literacy rate. So malaria gets entangled with poverty, with human well-being, with literacy, with demography, with social development. We just had a paper come out that shows in malarial regions, in this case in Italy and England, that it affects female age at first marriage, that societies that are under the pressure of malarial mortality have to reproduce faster, and the burden of this is really borne by women who have to get married earlier simply to keep a society reproducing itself. So it's a disease that really does more than just affect human health and life expectancy, but really grips the societies where it takes hold. And yet our knowledge of the history of this all-important disease is, to put it mildly, patchy. The written record is recent. It doesn't go back as far as the origins of the disease, and it's very difficult to use. I mean, Charles Dickens is not unusual in speaking of the ague, which is the old English term for intermittent fever. The Greek and Latin texts talk about intermittent fevers. We read about Corton and tertian fevers that describe fevers that come and go on a regular clock that's very indicative of malarial disease, but not necessarily completely diagnostic. So it's very hard to diagnose diseases from the kinds of texts that we have at our disposal. We can use human genetics, and there's certainly a long tradition of trying to understand the imprint of malarial disease on the human genome, leaving its traces in the sickle cell trait, the Duffy negative antigen, and so forth, that can provide clues about the history of the pressure that this disease has imposed on human populations. The phylogenetics of modern circulating malaria parasites give us insight into the ways that this pathogen has evolved. It's a pathogen that's really widespread in a number of different animals, especially primates, from which it moves into human populations. So we had pieces of the puzzle, but now aDNA is really filling in what we know about this disease and what we can know about this disease. And this paper is a huge step. I want to just highlight some of the major contributions from my perspective. And the first is methodological. I want to offer the authors my kudos for the quality, not just of the lab work and analysis, obviously, but of the history and archaeology that goes into this paper. The quality of any ancient DNA paper should obviously be judged primarily by its supplementary material. This is really where it's at. So I give you a little snapshot of it there. But as historians, we want to know. I want to know, who were these people? What do we know about the context of the individuals from which these samples were taken? It should be absolutely de rigueur in the field that there is not only site level but individual level detail on where samples are taken from, even on large scale studies. And this paper gives extraordinary detail. So my kudos to the authors for their methodological rigor. But this tells us that malaria spread early. There were suspicions of this. It may go back earlier still with more samples, but to me, seeing falciparum malaria in Europe in the site outside of Vienna going back into the first millennium BC is a big deal. We wondered if it was spreading into Europe this early. We had suspicions. Now we know. There's more to learn, but that's a big deal to see on the second box, Malaria falciparum, being pushed back in Europe into the first millennium BC. We see the key role of human mobility. As Megan said, it is weird to find Malaria falciparum in Belgium. That's unexpected. That's surprising. That gives us a challenge to think about. And this paper also tells us about the transmission of the disease to the Americans. As a historian who wrote a chapter about this just a few years ago, I can tell you that this really puts our knowledge on much firmer foundation. It's not, to me, surprising that the vivax malaria was transmitted from Europe into the Americas. In fact, not just to South America, but to Central and North America in European bodies that were infected with the parasite, and that the transatlantic slave trade played a crucial role in the transmission of Malaria falciparum. But I can tell you, as of a few years ago, this was based on a lot of guessing and inference, and now I think we can say we know. So this is a really remarkable contribution. At the same time, I'm just as excited about the questions that it opens up for the future. Can we refine our understanding of when it arrives in Europe? We need more evidence and more samples from Italy. Can we tell when it becomes endemic and when its presence is only sporadic? Can we tell that the disease is permanently endemic, or is it reintroduced with more samples? Say if we have a number of samples over centuries from the same location, we'll be able to identify whether the later lineages are directly descended from the earlier lineages in that location, or whether it represents the repeated reintroduction of this pathogen. I'll tell you, in having studied this disease, I'm very skeptical of the maps like the one that I showed that tell you where malaria was with these huge swaths of color. Malaria is a very regional, local, particular disease. The ecology of mosquitoes is absolutely crucial to the transmission dynamics, the ecology, and the distribution of the disease. So with this kind of evidence, and as we get more and more of it, we will have the ability to ask really penetrating geographic questions about what specific places this scourge took hold of in the human past. It was not equally widespread everywhere. Where I've studied it in England, it's in fact down to the parish, where the very local conditions of soil and elevation affect the ability of mosquitoes to breed an abundance that affects human health. So this opens the door to more profound geographic exploration. It lets us ask if the gaps are real, or are we going to see repeated reintroduction? Are we going to see establishment on a permanent basis? I want to know where the European strains of vivax come from. I know this paper can't answer every question, but it leaves us hungering to understand exactly when and exactly from where vivax was transmitted into European populations, into the Mediterranean, I should say. And finally, what opportunities lie ahead to consider both the history of the pathogen's DNA and the history of human population genetics. As has been said, malaria has left more and more evident traces in the human genome than virtually any other pathogen, certainly that we understand, in various red blood cell disorders particularly. And now we have the potential to ask questions about specifically the development of human genetic variation in response to this, the pressure of this terrible pathogen. So the future is very exciting. The future has been opened up by this paper. Thank you very much for this exciting contribution.
Let me invite our friend and colleague, Professor Dr. Ed Ryan, to come up and can we take the microphone?
Oh, I stand here, Mike. I don't have any slides.
Okay.
So wonderful, Dr. Michael, great job. So, and wonderful to be with you all today. So I guess I'll just touch base on a couple of big philosophical things that haven't already been touched on. And two little points. Number one is the CDC is in Atlanta because that's where malaria control efforts were first. And it was Mussolini who drained the swamps outside of Rome that broke the back of malaria just in the 1930s or in the peri-Roman area, et cetera. So this is an ancient disease. A couple of broad strokes. Number one is that malaria is a parasite, and parasites take the long game. So remember, humans any animal can get infected with either viruses, bacteria, fungi, or parasites. Parasites and fungi are eukaryotic organisms that are multi-chromosomed. And it's a long-term dance for parasites. Parasites literally are parasitos. They sit next to us. They sit at our table. So viruses like the herpes family or something might go deep in our body and become dormant and reactivate every now and then. TB can puddle along and reactivate, etc. But, plague tends to kill you, or measles tends to kill you, or then you're over it. Parasites take a totally different philosophy to life. They basically reach a detente with us, or any animal that they're infecting, and they're a constant infection. And so they are constantly evolving to try to get around our innate responses to try to bring them under control. We are constantly evolving to try to bring them under control, and it's a dance trying to get to transmission for each of us. And so the impact that parasites have on us is the long game. So that's why sickle cell has actually evolved at least 3 times, if not 5 to 8, based on haplotype analysis of humans. Hereditary spherocytosis, hereditary leptocytosis, G6PD deficiency, alpha thalassemia, what is thalassemia? It's the Greek word for thalatos, which is the ocean. It's also freedom, but the ocean, the sea, and that was the name because it was so endemic around the Mediterranean littoral. So alpha thalassemia, beta thalassemia, hemoglobin E in Asia, etc. Ancient, we've been fighting this for a long time. And this is not unique to us. We shouldn't be so anthropomorphic. Basically, every animal on the planet has its own malaria. So there's bird malaria, there's reptile malaria, there's human malaria, there's primate malaria, etc. We have 4 malaria species that evolved with us, and we are still fighting. There's a 5th knocking at the door, which is Nolsey, but the 4, and probably they become more attenuated with time, because there's no desire for them to kill us, right? They would love us to walk around like zombies, getting bitten by mosquitoes and going on for transmission. There's really no reason for them to kill us in 3 months or something, because they've limited the area under the curve they can achieve. And so, what else to say? So the 4 species, if you look at vivax and ovale, we call them the temperate malarias. And that's because we evolved in sub-Saharan Africa, and that's where malaria got introduced into us as a species. And gorilla malaria really doesn't jump into us, and we don't jump into gorillas or chimps or orangutans or anything like that. They're really species-specific because it's such a fine-tuned dance, not just between us and the parasite, but when there's a vector involved with mosquitoes. And it was alluded to, I think Dr. McCormick or Professor McCormick mentioned this. So remember, most of us don't even understand how complicated the vector world is. So under mosquitoes, we break them into major categories. There's the anophelines, the Aedes, the crudicine, etc. And then under the anophelines are the ones capable of transmitting malaria. There's over 500 species of Anopheles mosquitoes, about 30 to 60 that are competent at transmitting malaria. And they all have their own slightly breeding habits, inside, outside. How much do they like to take somebody else's meal, like a cow or a bird or something, versus us? So when you have these really complicated interactions among a triad, it's really hard to change that system. And when you do change the system, it comes out over eons, all right? So I suspect, based on no data, falciparum is the newest kid on the block, because he kills us at such a high rate. And vivax and ovale and malariae were probably much more ancient, and our détente with them has achieved something different. And the reason why vivax and ovale were selected for when we as a species walked out of Africa, or sailed out of Africa, or rode out of Africa, when we left there, we left places with year-round mosquito seasons, and we moved to places where the next mosquito might not bite you for 6 to 9 months. And so it's selected evolutionarily for a delayed release from the liver for what we call the extra erythrocytic life cycle in the hypnozoites hidden in the liver. So this is a really complex dance. What else to say? I'm really struck with the Habsburg data. I'm not surprised that Flanders was a hotbed for malaria because of its ecology. And what I am surprised of is that so many of the people, I think, from strata 2 and 3 had malaria. That really does suggest that, I don't know anything about the Bourbon House of Habsburg fighting in the lowlands, et cetera, for independence, or against independence for the lowlands. But it really does suggest that that's probably at a historic level a very interesting area to look at to see the impact. Because if you can take just random graves and find such a high impact of malaria on those individuals, I'd have to think that that affected your force reliability. And it may have led to the Protestants overturning the Catholic monarchs or something at that point. So, but I've waxed and waned, so happy to take any questions.
Thank you, Dr. Ryan. And so here to wrap up our conversation is the inimitable and admirable Professor Christina Warrener, who, as she comes to the podium and gets hooked up here, there's a microphone if you want to carry one around. Tina, here.
Okay, great. I think I'm okay.
Okay. Has been working on Science of Human Past for quite a while.
So I'm going to keep my remarks brief because I think we're a little bit over time, but I want to use my time to just pile on the praise for Megan, because I think this is such an extraordinary story, such an extraordinary effort. And so what I'd like to do is kind of put in frame kind of why this is so important and kind of where we've come over the last couple of decades. I think it's easy to forget that the field of ancient DNA is very young. The first ancient DNA paper dates to 1984. It's kind of amazing. I looked that up. That was the year before Microsoft Excel was made commercially available. It's an entirely different kind of universe. But the field kind of struggled to advance for a really long time because of technological limitations that we had in genetics. And it was really the transition to next generation sequencing and high throughput sequencing that had this incredibly transformative effect on our field in the early 2010s. And we didn't have the first pathogen genome, ancient pathogen genome, until 2011. It's really incredibly recent. There had been many attempts over a very long time to find malaria, and they had been met with repeated failure. It is a very difficult pathogen to find archaeologically. A lot of the early work that had been done had been trying to kind of modify clinical tests to look for specific epitopes and proteins using antibody binding. And they produced a lot of irreproducible results. And we know that's because now that proteins can break down in kind of unpredictable ways and will have kind of non-specific binding patterns that are unanticipated for these kind of assays. And so for a long time, the field was just really bogged down in this. And attempts to kind of find malaria more selectively using genetics were just not very successful. Part of the reason for that is that it's a quite difficult organism to study. As was mentioned earlier, it's a parasite. It's a eukaryote. It has a more complex genome. It's also really AT-rich, which might not seem relevant, but for those of you actually working in genetics, you know that makes things a lot more complicated. So DNA is composed of A's, T's, C's, and G's, and those C's and G's actually have a triple bond that hold them together, and whereas A's and T's only have a double bond. What that means, specifically in the context of ancient DNA, is that genomes that are rich in AT sequences fall apart more quickly. they're much more difficult to study, and they also typically have lower complexity, and there's all sorts of challenges in recovering them. And so when Megan embarked on this project, I have to admit, I was a little bit worried for her, given the many years of difficulty of people who had come. But she brought such a vigor and a verve to this, and really this just tenacious approach. And I was so impressed with at each challenge, she was coming up with creative solutions to overcome them. And so I was really happy to become involved in this project when she happened to find one of the malaria data sets in one of the data sets I generated from Nepal. And I was just as surprised as she was to find it. We had to do a lot of thinking as to why that might be, but the site is actually located in the Korla Pass, which is the lowest pass to the Himalayas. It's the only pass that's actually passable 12 months out of the year. It's long been a major trade route connecting the Tibetan Plateau and South Asia. And we know from funerary goods in many of these graves that they were actively trading further south in the Terai, which was endemic with malaria until very recently, but is fortunately, due to anti-malarial efforts at an international scale, now malaria-free. One of the things that I think is so exciting, kind of reflecting on this past, this kind of short history of ancient DNA, is that we now have, I recently did a tally for a paper I'm writing, there are over 10,000 human genomes that are now published, ancient human genomes. And in addition to the human DNA, you have all that kind of other DNA that's there, which may include the remains of pathogens that infected ancient peoples. We also have more than 600 ancient reconstructed pathogen genomes. This field has grown enormously. And I think it was one of the things I know that Megan kind of faced during this is with this kind of deluge of data, it became the kind of problems changed. Now, how do you manage so many data sets? How do you search so many data sets? How do you allocate enough computer resources to search so many data sets? And so I think she also became kind of earned an honorary degree in computer science along the way to try to solve these things. Just recently, Megan actually was a teaching fellow for a course that I teach, and I was actually just teaching it right before this, called Sick, 10,000 Years of Human Health and Disease, where we review what we know about the history of human disease, mainly through an ancient DNA lens. And while I was preparing for that class, I reread this journal that had been kind of recently published. It was actually originally written in 1544 by a chronicler who traveled with Bartolomé de las Casas, became Bishop of Chiapas, who traveled from Salamanca, Spain, to Chiapas in 1544, the year before the Great Cocolicli epidemic broke out. And one of the things I'd never noticed upon rereading is I'd always read the part where he arrived in the Americas and kind of all the diseases that they faced there, but I hadn't read the part where he was leaving Spain. And what really caught my attention as I was going through this again is they had this priest that was traveling with him. There were 46 priests, and one of them was named Domingo de Ara, and he had something called a 4-day fever. And they kept having to stop and leave him places and then come back and get him as they traveled because he kept having this 4-day fever. And then he eventually made his way on with them. And it only clicked at me at that point that, of course, he has malaria. And so I'm really excited. I think this is opening up so many new opportunities of unifying history, of taking a closer look at all of these particular accounts all over the world as we try to understand and reconstruct the history of this disease. So congratulations, Megan, on a really fantastic project that is opening up new doors and new imaginations.
Great. Really good. So thank you all for your presentations. Let me invite the panelists, now that Tina's got very comfortable there next to Dan Smale, to come back down and have a seat. And as we open the floor to the broader audience of interested, cultivated, learned public, some of whom are eminent specialists in many areas, including this one. It's a great joy. Our Science of the Human Past ambassador today, Emily Ding, who's been doing cool things in the Science of the Human Past herself as an undergraduate in the college, will help us by bringing you the microphone so that you can ask your question, and we'll ask you to say your name and to pose your question. We also have an online audience, and another supporter of the Science of the Human Past, Andy Chen, is working on that, so we may have questions coming in from around the globe. So, any comments on each other as we look for people who want to pose a question? I see that Professor Scott is leaping to the fore, and we'll have to share the microphones because we don't have enough.
Hi. Great. My name's Scott Edwards. I'm professor and chair of organismic and evolutionary biology. Amazing study, Megan. I'm so excited. And I will note you are a proud graduate of OEB 275, where we learned basic tools in population genetics. Congratulations. Yeah, so I'm curious, in the paper, was all the dating done with archaeological tools? In other words, did you ever have to use the actual sequences to date stuff? Is that even reliable? Is it complementary? Could that be helpful or yeah?
Yeah, this is on. So yes, all of the dating that we present in the paper is either from C14 dating or based on archaeological context. We did try some phylogenetic dating, so to try to leverage the power of ancient samples, the fact that we have time-stamped genomes from the past to learn about mutation rates of these organisms. We can then sort of use that rate information to try to back calculate the timing of particular divergence events. So for instance, when particular species emerged, that wasn't very successful, so it's buried in the supplementary material. It's complicated because these species have a mitochondrial genome, which is uniparentally inherited. It doesn't recombine, so it's kind of ideal for this type of analysis, but it's only 6,000 base pairs, and the diversity is just too low to really do a lot with. We didn't see any difference in the number of mutations in our samples that were 3,000 years old compared to mitochondria today. We also played around a little bit with the nuclear genomes, but they recombine really rapidly. So to try to use these approaches, which rely on sort of non-recombining regions of the genome, it was really complicated, unfortunately. So that's a big open question.
Hi, Irene Sotomarin. I'm in classics here at Harvard.
And I hope you're not going to react as happened to poor W.H.S. Jones.
No, not at all. Not at all.
I know you won't.
No. I'm excited. Just super. I work in Egypt, Roman Egypt. So my question is very basic. What kind of sample size do you need to test for malaria? Because in Egypt, for those of us that work, everything survives. Hair, nails, skin, bones. But the trick is, as Mike and I have spoken about this, is that very few labs, you can't take samples out of Egypt, and there's only one lab that you're allowed to run these tests to, maybe. So I'm always curious, I'm always asking anthropologists, what kind of sample size do you need? Can you do this on site? Do you need super specialized labs? Or because I'm a historian, so I don't know much about the science aspect.
Yeah, sure. So the sad truth about ancient DNA from samples with, preservation of soft tissue is that soft tissue just doesn't work very well. It doesn't seem that the DNA preserves very well in hair or, muscle or something. There seems to be something about the chemistry of bone that really helps with ancient DNA preservation. That said, we can still study, bones from Egyptian mummies or things like that. Although we do face a lot of trouble with contamination in Egyptian mummies, which is sort of a separate issue, but it's a big challenge. In terms of sample size, I guess more is better in a way, because we think that the preservation is maybe kind of rare, but also really stochastic. So to have a large sample size just gives you a better chance of identifying these organisms. But I would put a caveat on that this was sort of a first effort to pilot these capture reagents that we developed. So we picked the best ones, basically. And we didn't capture things that didn't show up in the computational screen that I did. So it's possible that we could be successful on other, samples that didn't show up in our screen as well. And I think we need to do some more work to evaluate what detection rates we'll have in samples that don't have sort of exceptional preservation.
Professor Higginbotham?
Thank you. I'm Evelyn Brooks Higginbotham. That was really very interesting. I'm interested in the sickle cell part of it. Because today, they have this now genetic modification that they're trying to do. But when I remember when we had the Catoctin furnace discussion. And when they looked at the aDNA for the skeletal remains, some of them showed this sickle cell, I guess, in there. So I'm interested in how this gets passed down, because if you look at, for example, the descendants, because remember they found that there were people in the Maryland area, actually a lot of the relatives, would they have the sickle cell trait? How does that evolve? Because this is well over 200 years we're talking about. And then I was also thinking about when the slave trade ends. So when you have, for example, in a place like Brazil, where you have so many more Africans, and it goes so much more, has anybody looked to see a comparative of just the sickle cell trait and what that might tell us about?
So you mean the sickle cell trait in populations like at Catoctin, where there's no ongoing burden from malaria, compared to places like Brazil, where malaria is still endemic today? That's a good question. I don't know too much about that, but I think, maybe someone else does.
You know, it'll, I don't know if this one is on.
Yeah, it's on.
It will depend on a fitness cost, right, of carrying the trait. And so since the sickle trait has very high penetrance and phenotype, the expectation is in the absence of malaria, it will disappear relatively quickly. And I think you see that in populations that are no longer affected by malaria. So, you know, the sickle trait is actually relatively uncommon in descendants from Africa in the U.S. I think the burden of disease in Brazil is quite low. So, I don't know, Marcia, would you agree? I think, whereas in Africa, you still see prevalence of 25% of the trait. So I think the right comparison is maybe Africa to places where there isn't high-level transmission.
And just to add to that, it's mostly vivax.
For the record.
Marcia Castro, professor and chair in global health and population, the School of Public Health. It's mostly vivax in Brazil, so the pressure is not there. So it's really impressive, the number of samples you use. So I have two questions. One is, if you could literally educate me when you say it's the first evidence of DNA. So when I was in grad school, I read that work from Salaris in the one child tested in the old cemetery, and it always was advertised as the first evidence of it was falciparum, if I'm not mistaken, in the bone of a child. So how is that different from your work? And I'm really trying to be educated here, because I always consider that as the first evidence of ancient malaria DNA. The second is, can you reflect of, I'm trying to get on the gaps we're going to have in this history, even if you can get more samples. So there are some places that culturally you have burials, so there's nothing left. And the second thing is, which is always a discussion in the Brazilian Amazon, that's where I work, it's very humid. So you can find remains of civilizations, but rarely you'll find bones that you can actually extract anything. So the more samples you get, you're still going to have holes in the map that you can't say anything because you have nothing to recover. So how that affects the history that you can tell us?
Yes. So to address the first question, I wouldn't claim that we have the first DNA evidence of malaria. What I think we can say is that we have the first genome-wide data. So the Salaris paper was a study that used PCR to extract these fragments of malaria DNA from bones. So looking for specific small fragments of the genome and then amplifying that many times to study that particular region or as a way of identifying species. What we're able to do with capture, which is new, is to reconstruct fragments from throughout the mitochondrial but also the nuclear genome And that gives us much greater resolution to try to compare the genomes of these ancient parasites to parasites that are still around today to make inferences about how they spread in the past and the evolutionary history of these parasites. To address your second question, that's a very important point. Yeah, there are regions in the past where we don't have burials, and we know that, unfortunately, for people who want to learn about the evolutionary history of malaria, precisely the places where malaria is worst today are the worst for ancient DNA preservation. We don't have great preservation in warm climates. We don't have great preservation in very humid climates. So that's going to be a major challenge. I don't know that I have an answer yet to how we can overcome that, except that I think maybe putting together different lines of evidence is the best that we can do. So looking at parasite genomes where we can recover them, but combining that with what we can infer from human genetics from the past, but also today in resistance mutations, and trying to leverage this combination of evidence to get at those hard to reach places.
Don Goldman, I'm an infectious disease college children, and also have a huge course on infectious diseases and social justice with Dr. Harper, and then I've got We showed a slide, I think, that showed a very strong correlation between literacy and malaria. I think it was in Italy. So, thinking about lessons from the past and the present, we know now, in the present time, that people who resonating. Better?
Yes.
Did you hear me all right, Dr. Harper?
Yeah.
Good. I tend to speak up. We know now that people who are impoverished and underserved tend to live in areas where they're exposed to environmental hazards, fence line communities, living in the bayou, in the swamp, whatever. Wouldn't that be the case also in Italy? So if you think who lived in the Pontine marshes, I don't suppose the wealthy Romans chose to live in the Pontine marshes. So can we extrapolate from at least reasonably recent times about those correlations and what might really be going on there? And then for Professor Worth, we're talking about genes. I'd love your opinion on genetically modified mosquitoes.
Yeah, it's a great question, and my belief is that that's a causal arrow that does run both directions, so that there's reason to believe, and people have worked on this. I mean, Jeffrey Sachs has worked on this a lot, where you try and find ways to isolate whether or not sort of the geography, the ecology of the disease is a truly fundamental exogenous first cause. And there's definitely some compelling evidence in some contexts that that's the case. That is, that the causal arrow goes from the disease environment to the social outcome. Mm-hmm. People who live in unhealthy environments have physiological, struggle to survive. They're less healthy. They have less time to invest in education. It lowers human well-being and these kinds of outcomes, of which literacy is just an example. But it absolutely is the case that the causal arrow goes the other way as well. That poverty causes infectious disease, that it increases the disease burden and makes for worse clinical outcomes in the case of infection. If you're poor, if you are food insecure, if you struggle for nutrition, your immune system doesn't have the fuel to fight infectious diseases. As you said, there's going to be selection for populations that can't choose where to live, that stay in the marshy areas. So I think there's good evidence on both sides of this, and certainly I'm someone who does not think that there's like one direction of that causal arrow. That's why I called it a correlation or an association, because I very much believe that those are entangled. And they can be, that's one of the reasons they can be very hard to solve, because you can't just come in with pesticides and nets and expect everything to be perfect, but at the same time, you need to get rid of the disease to create an environment where human societies can flourish.
Right, and there is evidence that malaria does lead to cognitive difficulty, particularly for children who go into coma and recover. So there's sort of, you could imagine a direct causal. And then to answer genetically modified mosquitoes, I think it's very exciting. You know, as Ed said, there are, you know, only 40 or 50 mosquito species that can transmit, Anopheles, that can transmit malaria. So one of the tricks is to try to convert those that can transmit into ones that can't transmit. That is probably, from an evolutionary sense, the safer mechanism. The other genetic engineering mechanism is to crash mosquito populations by essentially making sterile males or some other such methodology. The challenge there is you're going to have very strong selective pressure on survival. And so, you know, I expect there will be mosquitoes resistant to gene drive. So, you know, if you had to ask me which way to go, I would say trying to make mosquitoes refractory. Because all you have to do is interrupt transmission. You know, it's actually pretty easy from on paper standpoint. If I had a medicine I could give everyone that would knock down their parasitemia to a low level, I could do it today, and I could do it a month from now, malaria would be gone.
That's like Wolbachia.
Yeah, so Wolbachia is very effective for dengue, but minimal effect so far with Plasmodium.
We're going to have minimal time for continuing discussion, but we'll have one last question from the audience, and then someone who's participating globally who's waiting from South Africa. And with that, as usual, our thoughts and conversations have outstripped the time set for them in the formal moment, but we will continue informally in the reception in just a moment.
A quick question having to do with climate change and the spread of malaria and its rebounding. And it has to do with the fact that change is happening much faster than things are happening even genetically. That is, within decades, we're going to see 40 million people swamped and in search of fresh water throughout West Africa. And the result is the spread of the vector is going to be vastly different, and already has been in many parts of Africa, with it moving up in the mountains and the like. And the result is that malarial control is going to be separated from the history, in the sense of the evolution of the organism, the parasite itself, because it's going to have much more to do with the spread of the vector.
Oh yes, I think the spread of the vector has always been a very important part, and the viability of the vector, right, and the vector-parasite interaction. And that's why vector control has played such an outsized impact. So, you know, there are, all of this is based on modeling, but there is modeling to argue that 70 or 80% of the reduction in malaria from 2000 to 2015 was due to vector control. That's when insecticide-treated nets were widely distributed, these long-lasting nets. But it's also the time that rapid diagnostics and artemisinin combination therapy was distributed. So it's hard to completely untangle them. But I agree with you. I think the modeling for climate change, interestingly, there was both a Lancet and a WHO-based study. Both said, climate directly on the disease will go both ways. There'll be places where it's too hot for mosquitoes to survive. And breeding sites, Anopheles breed in small little puddles of water. Flooded areas will get rid of those breeding sites. So there are, this could go in several directions. It's unlikely to improve the situation, but it may not be disastrous.
And then one last question, which has come to us from our global audience from South Africa, that Andy Chen will share with us.
I'll go ahead and read it out. So we have a question from Calumet Links from Stellenbosch University in South Africa, an economic historian, and I'll essentially just read it. So they say, my question is, if we have enough samples, for instance, for Khoi or Maasai peoples, would we be able to trace movements and settlement patterns of ancient indigenous groups in Southern Africa based on disease burdens from ancient samples, just because these groups don't have any written history? So essentially, is population movement traceable through kind of pathogen history?
Go ahead.
I guess my sort of instinctual response to that question is that it could be interesting to put together studies of the human genetics and the pathogen genetics in contexts like that. So I have a lot of colleagues who work on tracing past human migrations using DNA from the humans themselves, and we know that that can be a really effective way of understanding how people moved across landscapes, and that if we can combine that with pathogen genomics, then it could be a way of trying to understand if disease was a driver of how people moved and why they moved to certain places in the past.
And your study of the Himalayan case shows, in fact, that individuals' mobility can be tracked through the pathogens that they bear. Yeah, okay, we can continue talking. We will continue talking in a moment outside, but first, a round of applause for Dr. Michael and for the wonderful colleagues who've spoken here. Yay.